Hi, I'm Sadashiva Pai from Science Mission. Today we have Dr. Alessandra Sacco with us. She is the Associate Professor, Aging and Regenerative Program, and Associate Dean of Curriculum at SBP Graduate School of Biomedical Sciences. She is from SBP San Diego, California. Dr. Sacco has won several awards. I'm going to name few of them. In 2017 WOW Award for Wonderful and Original Work Annual Retreat of SBP faculty. In 2014 again WOW Award for Wonderful and Original Work Annual Retreat of the SBMRI faculty. 2010 Cell paper selected from the Faculty of Thousand Medicine. 2008 Nature paper selected from the Faculty of Thousand Medicine. 2005 to 2008 MDA Research Development Grant. In addition, she has published more than 30 peer-reviewed articles, various book chapters, and she's in the editorial board of tons of journals, and she has received several grants for her work. Welcome, Dr. Sacco.
Hi, Sadashiva. Thank you for having me and for inviting me today.
Thanks for joining us. Let's talk about your background, education, etc. Give me a brief overview.
Sure. So I'm from Italy. I was born and raised in Rome, Italy, and I studied at La Sapienza University in Rome, and I completed my studies there, and in biology, in cell biology, and where there I studied the reactivation of the cell cycle in differentiated cells. So I was working while I was in Italy mainly in vitro cell culture system. And so I was interested in understanding how the differentiated state is actually can be regulated and can be also manipulated. Then after that, I decided to go for a postdoc abroad, and also because in Italy there are a lot of opportunities to grow in science, and so then I decided to come to the United States, and I did my postdoc with Ellen Blau at Stanford University. There, I studied skeletal muscle, but specifically, I started working with in vivo models and animal models, and that for me was really an awakening because I got closer to stem cell biology and really studying the world of regenerative medicine. So how can we actually repair a tissue and how a tissue is maintained? And so it brought me to ask the question that I'm still interested in studying today. And so there I work on skeletal muscle regeneration and specifically developing strategies to isolate muscle stem cells and identifying their potency, their ability to self-renew. And further, I work on developing a model for a muscle disease, which is Duchenne muscular dystrophy, which is progressive degenerative disease. And so we generated novel models that in many aspects, it's closer to the disease manifestation in patients. And so it's a useful model for testing therapeutics. I stayed there for several years, for about 8 years. So after my postdoc, I became a research associate and then senior scientist in Helen Blau's lab. And then in 2010, I came to San Diego and I started my own independent group at Sanford, Burnham, Prebys Medical Discovery Institute, which is an institute in La Jolla, which is a wonderful area for science because it's close to major institutions like UCSD, Salk, and Scripps. And here I established my own group where I continue to study skeletal muscle development and regeneration and understanding the signals that modulate stem cell behavior in this tissue and how can we leverage these signals to promote tissue repair and so maintain a healthy tissue function. So in terms of a little bit more about myself, I'm the first person in my family that went to university and graduated, and
Wow.
so and I owe this to really the efforts and sacrifices of my family, and I am
Mhm.
very grateful for them for allowing me to do that. So yes.
Good, good, nice, nice intro. And tell us about your lab people, grad student, postdoc, and all those things.
Sure. So my lab is, on average, we have 4 to 5 people, and it's a mix. Usually, it's a 50 postdocs and graduate students, and
Mhm.
then we have periodically also summer interns. So high school students that come here for the summer to experience science and the lab, and to see if that's a path that they might be interested in. And so currently we have one that it's just started for this summer. I like to have always a 50-50 between postdocs and students, because I'm also involved in the graduate program at our institute. So we started the graduate program at SBP that got accredited in 2015. So it's a
Mhm.
relatively new graduate program. And I'm involved in the program being the associate dean of curriculum. So I develop the courses and the educational activities.
Mhm.
And I find that it's very rewarding to be able to mentor trainees at different stages of their career. So postdocs, they already have a more clear vision of their career. So your role in that context is more like to help them to achieve their goal and then to move forward. Graduate student, the approach is a little bit different. It's really to help them to develop their own vision and what they want to do. And so a lot goes into having conversation and in trying to gather what actually they would like to develop and become in terms of professional career. And so I like to have this variety, because it really like allows me to have like a broader vision of mentoring younger scientists. And I have to say, like, mentoring is probably the most rewarding part of my profession. So I like that very much.
Yeah, you mentioned about the mentoring. You talked about it, and can you please tell more about your role models and how you got mentored and how you want to see it in your
Sure.
future?
So I had different experiences. So when I was in Italy, I was in a very small laboratory,
Mm,
and so the interaction that I had with my mentor was a daily interaction, and so it was a very close collaboration with my mentor.
mhm,
When I arrived in the United States, I entered a very big lab at Stanford University. There were over 20, 25 people.
mhm.
And so it took a little bit of adjustment. So, and I'm saying this because it happens to a lot of people that are doing this. So don't
Yeah.
be afraid. It's a normal part of the process and of the growth. And that helps you develop more your independence, because then you really find your own identity in a broader context at that point. You're not
Mm,
mentor every day, you're not that close to your mentor, but that allows you more to actually develop your own identity. I have to say that both of them have advantages and disadvantages. Overall, for me, experiencing both, it was actually extremely useful for my own growth. In terms of what I do in my lab, in terms of mentoring, I do have, given that it's a small lab, a daily interaction with them. But I do try as much as possible. So my goal is every day to try to empower them. So not really like stay on them and really like tell them what to do, but really it's more about help them asking the questions, and then give them space to find the answers and the approach to find the answers. And so sometimes, while at the beginning, when you start your own lab, this is difficult because you need to take a step back. Basically, you're not going to be the active role in every aspect of the experiment, but that's what I find it's most useful for trainees. In terms of role models, well, gosh, I mean, there are a lot of role models in my life. I have to say, from a personal point of view, the role models for me have been strong women in my life, and so really like my grandmother and my mother, that they face a lot of adversities, and for me, the value really of a role model is the everyday, facing the everyday challenges. Is not really like necessarily the big achievement, but really facing with dignity and strength the everyday challenges. And I find that like an incredible strength in that. Um, in terms of um, in terms of scientific role models, for me, I am very, I have a lot of appreciation for women who became faculty decades ago, like in the late 70s and 80s, because it was really difficult at that time for women to enter the world of academia. And they really open up the path for a lot of us moving forward. And so for them, facing this adversity and breaking basically and to open that path was actually for me is extremely educational for that point of view. So yes.
And like you talked a lot about your lab and all. Like let's just briefly talk about other than lab. Like what are your hobbies and what else do you do other than research?
All right. So I love animals, and so I have cats, and I like a lot to. I like to spend a lot of time with them, and so that's one part of my life. But then being in San Diego, of course, I love sun. I love the sun, and I love the great weather. We're very lucky here, and so I like to be outside in the sun for most of the time. I have to say. And then on a more personal note, I'm interested in psychology. So I'm really interested in understanding psychological behaviors, which I think it's kind of a personal interest for me, but it helps me sometimes because even in my daily work, because um, one thing that at the beginning, when you are starting your own lab, find difficult, at least it was difficult for me at the beginning, is really to realize and accept that different people have different needs. And so you need to approach them differently to help them to achieve their goal. And so again, being interested in the psychological aspect of it helps me a lot to be able to try every day to do my best to help each person with their own needs. Yeah.
That's interesting. So I haven't heard this answer from anybody. No, it is, as you said, it is important because the grad student postdocs, their needs are different and individually. And now in the labs, like you have people from different parts of the world. So they are all different. They come with their own way of thinking and all those things. So that's really useful.
Yes.
So now going back to the research, like you mentioned about the stem cells, muscles, and all those things. So you can give a small overview and start your presentation.
Sure. So, as I mentioned, so we study skeletal muscle, and we are particularly interested in stem cell biology within skeletal muscle. And the interest for me in skeletal muscle comes from the fact that it's an essential tissue, and it's affected in a variety of human diseases. So not only diseases that specifically affect muscle, but also diseases that affect other tissues, but they have an indirect effect on muscle. The results of having a reduced muscle function is actually extremely invalidating for a lot of human diseases, and it really like reduce the quality of life, and in some cases it shortened the lifespan of patients. So there is an incredible interest in trying to develop approaches to promote skeletal muscle repair, because that would have an impact not only in muscle degenerative diseases or in the context of aging, where we know that skeletal muscle progressively decrease their function, but also in a different variety of contexts, like, for example, even in cancer, where muscle mass is progressively reduced, for example, in cachexia. So, trying to understand how the muscle can be repaired and how can we maintain a healthy function of muscle is a central question that we have. And we leverage stem cell biology and stem cells to try to utilize their potency to achieve these goals. So that's the main aspect, the main framework in which we actually um, perform our research. So Would you like me to start my presentation now?
Yeah, sure.
All right, so let's do this. Let me just do this. There you go. All right, can you see it?
Yeah.
Perfect. Okay, so I'm going to give a brief overview, and then I'm going to discuss an example of how we can actually manipulate muscle stem cells and promote skeletal muscle. Feel free to interrupt me if there is
Yeah.
any question that you have.
Okay.
Okay, so we study skeletal muscle, and that actually is an essential tissue for voluntary movement, as we know, to actually be able to walk and also to move and also to breathe. So it's a fundamental tissue that we have for our life. There are over 600 muscle groups in the human body, and skeletal muscle is also has an incredible ability to adapt. And we can think about, for example, athlete that runs marathons versus athlete that runs 100 meters. They have very different adaptation into how skeletal muscle perform. So has a strong ability to adapt to its use that we actually make of it. The basic unit of the skeletal muscle is the myofiber, which is a very unique cell because it's a multinucleated cell. It's a syncytia that is generated by the fusion of myogenic progenitors with each other to form this giant cell that contains all the structural proteins that are required for contraction and hence movement of our body. Now, skeletal muscle has a remarkable regenerative potential. So if we have an acute injury, for example, or even if we have like a strong exercise and we can have mild damage to the muscle, it's able to repair itself. And this is due to the presence within the tissue of tissue-specific stem cells that are called skeletal muscle stem cells. As you can see in this slide on the left, this is the first picture of a muscle stem cells that was published by Alexander Mauro in 1961. It's an electron microscopy picture. You can see these muscle stem cells that is basically adjacent to the muscle fibers and underneath the basal lamina that surrounds each muscle fibers. And remarkably, already at that time, the author hypothesized that these cells could actually be remnant of embryonic origin that could be deployed again to repair the tissue in case of a damage. And indeed, this turned out to be true. It was a remarkable description at the beginning, but it turned out to be true. So now, several decades later, what we know, as you can see on the right, is that muscle stem cells are present in the tissue, and they're largely in a quiescent state. Now, quiescence is a state in which a cell is not dividing, but is able to respond to signals, and it can be stimulated in response to signals. And so in response to stress or in response to injury, is able to activate and undergo proliferation. And through proliferation, it can actually generate more committed progenitors that are called myoblasts. And that ultimately they will divide and they will differentiate and fuse to repair the damaged myofibers. Now, throughout this process, of course, through the division process, muscle stem cells not only give rise to myogenic progenitors, but also to more copies of themselves. And this process is called self-renewal. And this is a fundamental process because it ensures that the number of muscle stem cells in a tissue is maintained constant. And so after an injury, you do not lose muscle stem cells, but they are maintained and they are ready to be able to respond to the next round of injury. So over the years, specific transcription factors have been identified that mark these distinct stages. So muscle stem cells, as you can see on the graph on the scheme on the right, are marked by the expression of PAX7. Myoblasts, or committed progenitors, are marked by the expression of MyoD. And when they differentiate and exit the cell cycle, they are marked by the expression of myogenin. And then throughout differentiation, they will start expressing all the contractile proteins like the myosins, for example, that are essential. So, and these markers are very useful for us to be able to observe and monitor the myogenic progression of muscle stem cells, both in a tissue and also ex vivo if we perform in culture type of assays. Now, I mentioned the skeletal muscle as a very strong regenerative potential. However, this is not always true. There are chronic conditions in which the ability of the tissue to repair itself is compromised. And this is, and I'm going to provide a couple of examples, chronic degenerative diseases, like, for example, muscular dystrophies, in which the constant damage of the tissue leads over time to the exhaustion of muscle stem cell function, and also in the context of aging. So during aging, we do observe maybe the most known manifestation of skeletal muscle in aging is sarcopenia. So it's a progressive reduction of our muscle mass. However, what we know is also that if in age context we actually have an injury, the ability to repair our tissue is actually much slower and in some cases is compromised. And this is due to a dysfunction in the muscle stem cell compartment. And over the years, and mostly in the most recent years, there have been, like, as you can see in this slide, several specific processes that have been identified as deregulated in the context of degenerative diseases and aging. The first one is the loss of quiescence and self-renewal. So upon an injury in this chronic context, the cells is going to have a defect in self-renewing, so in generating more copies of itself. And this progressively is going to lead to a reduction in the number of stem cells, and so in exhaustion of the muscle stem cell pool. A second aspect is increased myogenic commitment. So upon cell division, muscle stem cell in this context can be more biased towards becoming progenitors at the expenses of self-renewal. And again, the outcome of this is going to be a reduced number of muscle stem cells in a tissue. They can also exhibit an aberrant differentiation. Now, muscle stem cells are cells that make muscle, they're tissue specific. However, it has been shown that for example, in aging context, muscle stem cell can also give rise to fibroblasts, so adopt a different phenotype. And this is going to be, of course, at the expenses of a healthy repair of the tissue. And the fourth one is cellular senescence. Cellular senescence is a biological condition in which the cells are no longer dividing, and so they can no longer respond to stimuli and repair the tissue properly. But the intriguing aspect that has been emerging in the last few years is that in senescent cells, they not only lose their ability to contribute to the tissue, but they also have a deleterious function in the tissue because they secrete a variety of factors that negatively impact the microenvironment. So they have a dual negative effect. They are no longer contributing, but they also have a detrimental effect on the microenvironment. So these are four processes that we know that they are actually contributing to muscle stem cell dysfunction in chronic degenerative diseases and aging. So what we're interested in studying in our lab is what are the signals and what are the molecular networks that regulate muscle stem cell behavior, and how can we manipulate these signals to promote expansion of muscle stem cells and then efficient repair of the tissue. So what we study here just put two main areas that we're interested in studying in our lab. The first one is how the muscle stem cell interact with the microenvironment. So in the muscle tissue, as you can see on the schematic on the left, of course, the cells are not in a vacuum. They're actually able to interact with different cell types. They're very close to the myofibers, which is the mature skeletal muscle cells, but they're also very close to blood vessels, to nerves, fibroblasts, also adipocytes, so fat cells. So they are exposed to a variety of different cell types and a variety of signals that are emanating from these cell types. And what is becoming more and more appreciated in the recent years is that the signals of these different cell types are very important to coordinate temporally tissue repair. So really understanding how muscle stem cell receive this signal and how it translates this signal in specific different gene expression is very important for tissue repair. So one pathway that I'll talk about today that we're interested in, as you can see in this slide, is the JAK-STAT signaling pathway. Now, JAK-STAT signaling pathway is very important in mediating the signals of several secreted components. For example, interleukin-6 and oncostatin M, to cite only a couple of them. And upon binding of these secreted factors with their receptors, JAK2, as you can see here, is a kinase, phosphorylates STAT3, and in tyrosine 705. And upon these phosphorylations, STAT3 can dimerize and then translocate to the nucleus, and being a transcription factor is going to regulate transcription of a variety of genes in muscle cells. And I'm going to talk about a little bit what is the function of this signaling pathway in muscle stem cells and how is important for cell fate choices in muscle stem cells. Then a second aspect that I won't talk about today, but is actually extremely important and is becoming like a major question in stem cell biology, is understand the heterogeneity of the muscle stem cell compartment. So in a population of cells, not all cells are equal. They're very different. They have different potencies, and they have different expression of genes. And these can be due to their own autonomous differences, but also it can be due to the local signals to which they're exposed to. And really studying and trying to understand the the composition of this compartment and how the presence of all these different subpopulation is important for maintaining a healthy tissue is a question that we are interested in studying mostly in vivo with lineage tracing and clonal tracing in vivo. But today I'll focus on the signals and specifically JAK-STAT3 signal pathway that we've been studying for the last few years. So what we have shown a few years ago, this is a paper published in Nature Medicine 2014, and that was done at the same time also with Michael Rudnicki's group that published also similar findings, is that upon activation of quiescent muscle stem cells, we have activation of STAT3 signaling. And STAT3 signaling is very important to promote the transition of the cells to a committed stage, so to committed progenitors. And so it allows the progression of muscle stem cells through myogenesis, so from the quiescence and activated stage to differentiation and then mature myofibers. This pathway has been mostly studied in culture in the past years, but here we were actually studying in vivo in the context of in vivo animal models. And so what we have shown, I'm just showing a couple of results from that paper because it was published, is that given that STAT3 signaling is promoting this commitment of the cells to the progenitor stage, the question was, can we manipulate these signals? Can we transiently inhibit STAT3 activity? And would that result in an expansion of muscle stem cells? And then after we remove the inhibitor, now we have a larger pool of stem cells that can contribute more efficiently to the tissue. And this turned out to be true. So what we did is that we took a mouse and we induce an injury, and with, in this case, with notexin, but which is a snake venom toxin that induce massive, significant tissue damage. But there are a variety of different ways to induce localized muscle injury. And then after this, we injected into the muscle a STAT3 inhibitor that blocks the activity of STAT3. And then after that, basically, we monitor the activity of muscle stem cells and how the tissue was repairing itself. And as you can see here, so when we did this in young mice at 2 months of age, this is an histology, it's a cross-section of a muscle. So all these round pink circles are actually muscle fibers. You can see that in muscles injected with the STAT3 inhibitors, the fibers are bigger. And this is due to the fact that, I'm not showing it here, that we actually expanded muscle stem cells that then contribute more efficiently to tissue repair. And very interesting, what we also observe is that the beneficial effect was also true when we induce an injury in an old mouse at 24 months of age, which is an age mouse. We also observe, as you can see on the right, larger myofibers that you can see here when we inject the STAT3 inhibitors. So this is one example, and so if you transiently inhibit a signal that promotes the transition of muscle stem cell to the progenitor stage, you can basically allow the expansion of the cells, and then these cells are in larger numbers and are able to repair the tissue more effectively. So this was done in the context of young and aged, but we also tested it in the context of a chronic degenerative disease, which is Duchenne muscular dystrophy. Now, as I mentioned, Duchenne muscular dystrophy is a chronic degenerative disease due to mutation in the dystrophin gene, and that causes a constant damage to the muscle. And so over time, the ability of muscle stem cells to repair the tissue becomes compromised. Now, the mouse model that we use here is the MDX mouse, which has a defect, as a mutation in the dystrophin gene. As you can see on the top left, this is a Western blot that shows that in dystrophic conditions, we have an elevated phosphorylation of STAT3. So the pathway is constantly and aberrantly activated because the muscle is always damaged. And so these results, we hypothesize, results in basically a premature commitment of muscle stem cells. So we repeated the same experiment that we did for the young and aged mice. In this case, we took a dystrophic mouse, in this case, a more severe models of dystrophy, the MDX-MTR. We injected the inhibitor in the muscle for STAT3 every week. And then after 1 month, we look at the muscle stem cells. And you can see in the bottom left that indeed muscle stem cells, also called satellite cells, were more proliferative in the presence of the STAT3 inhibitors because we are blocking their commitment and differentiation. And if we look at the muscle fibers, you can see that mice muscles injected with the STAT3 inhibitors have larger myofibers, and was also measured on the top right, as you can see here. So again, manipulating this pathway with inhibitors, it allows us to expand the cells and then promote tissue repair. And there are a lot of inhibitors of the JAK-STAT signaling pathway because this pathway is involved also in several types of human cancers. So there has been a lot of interest in developing different, from pharmaceutical companies in developing different inhibitors. And so one aspect that we're developing now is actually testing these specific inhibitors and trying to see basically also the long-term effect of these inhibitors in tissue repair. But of course, I mean, the question after this proof of concept that actually we can manipulate a signal that promotes this transition and as a result improve tissue repair is what are the downstream effectors of STAT3 signaling in muscle stem cells? So what are the genes that are regulated transcriptionally by STAT3? And what can we learn about how is actually promoting this commitment towards myogenic progenitors? And so to do this, what we did in the lab is actually we started by utilizing a genetic model in which we can delete STAT3 specifically in muscle stem cells. So in this case, we utilize, as you can see the scheme on the top, a PAC7 CRIAR. So it's basically a model in which you can delete STAT3 gene specifically when you inject the mice with tamoxifen, and specifically in muscle stem cells, because the recombinase CRI is expressed under the promoter of PAC7, which is specific for muscle stem cells. And so we delete the gene, specifically muscle stem cells, and then we induce an injury. and then we isolate the cells from the tissue, and we perform RNA sequencing, RNA-seq, to see what are the differences in gene expression in the presence or absence of STAT3. And when we did this, we have done this both in healthy tissue or in tissue that were regenerating. And you see the heat map. There were significant differences that you can see on the left. And on the bottom right, you can see that when we look at control mice and we look at the difference between regenerating muscles and healthy muscles, you can see that the pathways, the processes that are mostly affected are proliferating genes, because the cells enter the cell cycle and start dividing. Protein synthesis, because the cells start to actually increase their synthesis, the rate of protein synthesis, and also mitochondrial function, because during the activation process, the cells undergo metabolic reprogramming. They shift the way that actually they produce energy, and they increase the mitochondrial respiration. But interestingly, if you look at cells, muscle stem cells in which STAT3 was ablated, you can see that the mitochondrial function process was no longer modulated. So you can see proliferation changes and protein synthesis changes, but there were no longer the metabolic reprogramming happening like in healthy tissue. And so that led us to hypothesize the STAT3 pathway is actually promoting mitochondrial function during muscle stem cell activation. To gather a little bit more evidence about that, we actually isolate the cells. And as you can see on the top, we sort the cells. And there is an example of a FACS flow cytometry plot in which we can isolate the cells based on cell surface markers, CD34 and alpha-7 integrin. And then we place the cell in culture. And then we measure with the seahorse assay the oxygen consumption rate, the OCR, in muscle stem cells. And you can see, as you can see in the bottom of the slide, in the muscle stem cells in which STAT3 is deleted, we have a reduced mitochondrial respiration, OCR, both in basal and the maximal conditions, while actually the glycolysis measured with the extracellular acidification rate on the bottom right was not affected. So really STAT3 signaling is actually promoting in muscle stem cells mitochondrial respiration. But how does it do that? So what are the genes that are involved in this process? And one reason why we are particularly interested in this is because it was becoming more and more evident in the recent years. There have been several studies really interested in understanding how the changes in metabolism in muscle stem cells, but this is in general also in stem cells from other tissues, are actually important to direct cell fate changes. And so regulation of stem cell metabolism is actually very important to determine what the cells will become after. So we focus on the genes, as you can see here, in terms of understanding the downstream effectors of STAT3 in genes that change when during the activation process. You can see the 63 genes that is actually highlighted here. So the change during activation in healthy tissue, and they are no longer responsive in the knockout for STAT3. And so we tested several of these genes, and one of the genes that we became interested in was FAM3A. So FAM3A is a ubiquitously expressed cytokine-like protein, and it has been shown, although it's not been heavily studied yet, but it's been shown in vitro to induce mitochondrial respiration in vascular cells, neuronal cells, and hepatocytes. And so, of course, I mean, there was an interest that this gene might be involved in the phenotype that we are observing in the absence of STAT3 signaling. So we validated that FAM3A is indeed downregulated in muscle stem cells without STAT3, as you can see in the bottom right. And also, when we isolate muscle stem cells and we induce differentiation into myotubes, so differentiated muscle cells in vitro, And that's a very easy assay that you do basically with serum starvation. So you reduce the nutrients in the media by reducing the amount of serum, and the cells spontaneously will differentiate into mature myodules. As you can see, FAM3A is actually at the RNA level upregulated during differentiation, and that is very similar to what happens with STAT3, that it also becomes upregulated during differentiation. So they have the same trend in terms of RNA levels during the differentiation process. But of course, the next question was, well, is this a direct target? I mean, during RNA-seq, we isolated several genes, but is it directly regulated by STAT3? So we, by bioinformatic approaches, identified a putative binding site of STAT3 on the promoter of FAM3A, as you can see in the top left here. And so when we took a myogenic cell line, in this case C2C12, and we actually kept it in growth conditions, and we also stimulated with IL-6, interleukin-6, that stimulates STAT3 signaling. As you can see here, when we stimulate the cells with STAT3, this is a chromatin immunoprecipitation assay, a chip, we can see that actually the STAT3 occupancy on the FAM3A promoter is increased. And that was actually correlating with an increase in the H3K27 acetylation mark, which is a mark of active transcription. So this indicated that FAM3A is a direct transcriptional target of STAT3. Further, to further substantiate this, we look at previously published RNA-seq data, and we observe, as you can see in the top right, that in correspondence of the transcription start site of FAM3A, we have a peak of MyoD binding. Now, MyoD is a major transcription factor in myogenesis that is essential for the myogenic transition of muscle stem cells towards more mature cells. And so we observed this in C2C12 in previously published data, and also in ChIP-seq data of our collaborator, Lorenzo Puri, when he actually convert fibroblast into myoblast by overexpression of MyoD, we can see again also in this case that MyoD is actually binding in the correspondence of the start site of FAM3A. So this was interesting to us because we are seeing that STAT3 is regulating FAM3A, but also MyoD, which is an essential transcription factor for myogenesis, is regulating FAM3A. So, and this would actually point to a network loop in which both STAT3 and MyoD regulate the transcription of a gene that might be involved in metabolic regulation of muscle stem cells. Now, on the bottom left, you can see that when we isolated muscle stem cells from mice lucky MyoD, this is a genetic model lucky MyoD, the FAM3A RNA levels are reduced, again, consistent with the fact that the gene is regulated, positive regulated by MyoD. And finally, we actually perform a luciferase assay. This is our collaborator, Lucia Latella in Italy, and in which, when we have a construct in which we put the FAM3A promoter regulating luciferase, and we co-express in 293 cells also STAT3 and MyoD. And you can see that when we actually overexpress STAT3 and MyoD, we do have an increase in the luciferase reporter activity for FAM3A, indicating that, again, STAT3 and MyoD are upstream regulators of FAM3A gene transcription. So moving forward, so this was interesting to us. So we identified a gene that is actually regulated by STAT3 and by MyoD, and has been previously implicated in regulating mitochondrial function in different cell types. So now the next question was for us to try to understand what happens when we manipulate FAM3A. So here we perform an acute loss of function. So we isolate muscle stem cells and we infect them with a lentivirus that expresses an shRNA against FAM3A to block its expression, or an SH control. As you can see in the top right, this is at the RNA level. You see that when you actually inhibit FAM3A, you have a downregulation of myogenin, which is a marker of myogenic differentiation, and which is again consistent with the fact that if you block FAM3A, you block that transition towards later stages of myogenesis. And also we look at the protein level when we actually knock down FAM3A in muscle stem cells. This is in culture. at 72 hours in growth condition in the bottom aspect, in the bottom side of the slide. You can see that when we inhibit FAM3A, we do see an expansion of cells that are expressing PAC7. This is the dark gray and the black. And so this was suggesting, again, consistent with what we observe with STAT3 gene deletion. So if you block FAM3A, you are actually increasing the number of cells that are less committed and blocking the transition towards more committed stages. But this was done in vitro in acute loss of function. So the next question besides here is just a validation also that this results in an inhibition of expression of myogenin still in vitro and an inhibition of differentiation. So the formation of multinucleated myotubes expressed in myosin heavy chain. Again, validating that this is a gene that is required for proper myogenic differentiation. So the next question for us was to test it in vivo, because the acute loss of function in vitro, of course, it's useful, but has the limitation of is it actually happening? Is it a process happening in vivo? So what we did, we generated a knockout mouse model, the deletion of FAM3A using CRISPR-Cas9, and then we look at the skeletal muscle, both at fetal stages and postnatally. So here you can see that this is at fetal stages at embryonic E15. You can see that in green there is embryonic myosin heavy chain that marks muscle fibers, and in red there is laminin. You can see that you have a reduction of the size of the muscle. These are the limbs, the lower limbs of the embryo. So you have a reduction of muscle mass. And if you look at the bottom side of the slides, you can see that this resulted in a reduction on the number of myogenin positive nuclei. Again, very similar to what we observe in vitro. So deletion of FAM3A is impairing the ability of muscle stem cells to transition to committed progenitors, and then this results in a smaller muscle mass in the embryo. And this was also true at birth at P0, as you can see here on the left, again, a reduction on the area, so the muscle size expressing embryonic myosin heavy chain. And on the right, you can see that this is actually was correlating with an increase in the number of PAC7 positive muscle stem cells. So we are able, if you delete the gene also in vivo, we are able to increase the number of muscle stem cells at the expenses of the differentiation, and so resulting in a smaller muscle mass. So this is done at fetal and natal stages, but now we also look at the mouse line in the adult. And as you can see here, the body weight in the top left is reduced in these mice, and this results in the reduction of the muscle mass in several muscle groups. You can see the gastrocnemius and the quadriceps muscles that are reduced in mass. Interestingly, there is a significant reduction in the heart, and we're exploring that to study the function of this gene also in the heart, while there was no changes in the other tissues. So really the reduction in muscle mass was related to the reduction of, the reduction in body weight was related to the reduction in muscle mass. So here, so we saw so far what the deletion of the gene can do to muscle stem cells and to the embryonic development of the tissue and in the adult. But what is the function? How does this protein work? Now, the protein is predicted to be secreted, and so we wanted to test that. So we took C2C12 and we overexpressed FAM3A tagged with a MIG tag here. You can see on the left that the protein is colocalizing here with the ER in the cell. And if we use an inhibitor of secretion, which is monensin, you can see that the protein accumulates inside the cells, is again in the ER, again indicating that the protein is being actively secreted by the cells. This was quantified, I'm not showing here in terms of colocalization, but further we also did Western blot. And you can see here on the top right that when we actually block the secretion with monensin, we have an accumulation of the flag FAM3A protein in the cell, as measured here. And finally, in the bottom right of the slides, you can see we took the media And then we did a Western blot in the media of the transfected cells, and we do see that the FAM3A that we transfected, the protein is present in the media. So all of this evidence together is actually validating that this protein is being actively secreted by myogenic cells. But of course, being a secreted factors, there is always the question in the microenvironment that is it only being secreted by muscle cells or by other cell types? So we really wanted to see where this signal is coming from in the muscle microenvironment. And so we isolated both in pups and in the adult. You can see in the left side of these slides, we isolated by flow cytometry muscle stem cells, macrophages marked as MPs, fibroadipogenic progenitors, which is a very important cell types that can adopt a fibrogenic or an adipogenic fate, but is very important for signaling muscle stem cells, and also myofibers. And you can see FAM3A is being expressed by all these different cell types, with the majority of it being expressed by the myofibers. And when we induce an injury in adult tissue, you can see that we can have, and then we look at 3 and 7 days after injury, you can see in the right side of the slides that we have a transient expression of this gene in several of these cell types, specifically in fibroadipogenic progenitors and macrophages, really indicating that both during development and during regeneration, and the adult regeneration in several aspects recapitulates the development of the tissue. It reactivates several signals that are actually present during developmental myogenesis. You do see a transient activation of this protein. So this protein is produced by several of these specific cell types. And so then we decided to take advantage of the function of this protein that is actually secreted. And so we exposed muscle stem cells, either healthy or lacking STAT3, to a recombinant FAM3A protein. So we put the protein in the media, and then we ask, can basically the exposure of the cells to this protein rescue the phenotype of the cells that are lacking STAT3? So that was the question, to understand what is the relevance of this gene as a target of STAT3A. Is it relevant for the phenotype that we observe? And indeed, this is the case, because as you can see in the top right, when we expose the cells to FAM3A, you can see, look at the graph, that in the STAT3 knockout cells in the black bar, they have, in non-treated conditions, they have a reduction on the percentage of myogenin positive cells, because STAT3 is required for myogenesis. But when we expose these cells to FAM3A, we have a rescue in the ability of these cells to express myogenin. So again, indicating that this gene is relevant for the phenotype that we observe with STAT3 knockout cells. Further, this protein, the exposure to the recombinant protein, was able also to rescue the metabolic phenotype, the mitochondrial respiration, because upon exposure to FAM3A, we do see the muscle stem cells now rescue their ability to have proper mitochondrial function. And again, the glycolysis was not affected. So it's able to rescue the ability of the cells to commit towards differentiation and to restore mitochondrial respiration. This is done in vitro. Then finally, we decided to test this in vivo. And so we took the mouse model in which we delete STAT3 specifically in muscle stem cells. We induce the gene deletion with tamoxifen treatment. We induce an injury of skeletal muscle, in this case with barium chloride. And then we inject intramuscularly the FAM3A recombinant protein. And then we ask, 6 days after injury, is the muscle regenerating more effectively, and are muscle stem cells any different in the presence of this recombinant protein? So as you can see at the center of the slide, when we look at the STAT3 knockout cells, these are the black bars. In normal conditions, they have an increased number of PAX7 positive cells, because the cells are accumulating in this uncommitted state. And when we actually inject FAM3A, now we are again normalizing the numbers of PAX7 cells in the tissue. And if you look at the bottom part of the slides, if we actually look at myogenin, we see the same thing. So the STAT3 knockout cells have a defect. This is the black bar in transition into a myogenin positive stage. But if we inject the recombinant protein, now we are rescuing this ability to actually transition to this stage and make muscle. So altogether, what this story that was recently published a couple of months ago is actually telling us that upon the activation of muscle stem cells, the STAT3 signaling pathway is promoting the transcription of FAM3A, which is a secreted protein, not only by muscle cells, but also from other cells in the microenvironment. And this FAM3A is promoting mitochondrial function, and this promotes the commitment of these cells toward differentiating progenitors and then making muscle. And of course, what we're interested in now is actually utilizing this as a potentially even more specific target compared to STAT3, and trying to see whether if we can use approaches to block it, and it can be like chemical compounds or antibodies, if we can actually have in chronic conditions where actually STAT3 is chronically elevated and this protein is chronically elevated, if we block its function, can we actually now restore and promote healthy repair of the tissue? This is the conclusion for this part of the story. And so what emerged from this study is that STAT3 promotes mitochondrial function in muscle stem cells, activates the expression of FAM3A, and FAM3A promotes muscle stem cell myogenic differentiation. And if we delete the gene in vivo, is impairing muscle development. And FAM3A is a relevant target of STAT3 because when we actually expose the cells of FAM3A, cells that are lacking STAT3 is able to rescue the phenotype that we observe. So this is a story that tells us, that sheds some light on what is the biological process that STAT3 is affecting in muscle stem cells, which is mitochondrial function, and identified one gene that is actually relevant for this specific phenotype. We have many more that we're now testing, of course, because STAT3 activates, regulates several different genes, and so really trying to get at the networks that are actually regulated by this pathway, and what can we learn about these networks also in chronic conditions. So I think I'll stop here. I just would like to thank the people who did the work, which is in my laboratory. Former members Tufan Aidogdu and Matthew Tierney did the STAT3 story in the genetic deletion in the mice. David Sala did the specific studies on FAM3A, and but several people in the laboratory collaborated with these stories. Our collaborator Pier Lorenzo Puri and Greg Duster for the genetic deletion of FAM3A, and several others, and our funding sources. And just would like to take this opportunity just to say that we're also, I'm one of the organizers of the Frontiers in Myogenesis conferences that will take place in September, this September in 2019 in Costa Rica. And we have an outstanding lineup of speakers. And so I just wanted to put that out there. Whoever is interested, the website link is there and the email is there. And it's going to be a very interesting skeletal muscle meeting. So that concludes my presentation, and I'm happy to answer questions. I'm just going to go back here and stop the screen sharing thing. Let's see if it works. Eccoci.
Here you go. Thank you so much. It's an interesting work. Very, very nice work. So I have a question.
Sure.
On FAM3A, right?
Yeah.
So in your model, you had mentioned that FAM3A is secreted, and again, it acts on the same cell kind of thing. And at the mitochondrial level, whatever it activates mitochondrial function or whatever.
Yes.
Like, do you have any receptors for the FAM3 on the surface?
That's a great question. So the receptor is not known, and we are looking at that actively right now. So we are utilizing approaches, proteomics approaches, to actually identify cell surface molecules that are binding to FAM3A, because really that's a relevant question. So not only the signal, but how are the cells responding, and is the receptor modulated in chronic conditions? That's a fantastic question. So we're studying that right now. Yeah.
Okay. And the other question is FAM3 acts on these cells, right? Whatever stem cells.
Yes.
So now, like, there should be a off-target effect on FAM3A. In addition to that, because in order for the the cells to grow to form myofibril, you need the vasculature. There
Yes.
needs to be like blood vessels. Does it have any effect on other cells, like vasculature or any other cells?
Yes, we are starting to look at that right now. I agree. Every time we introduce something in a tissue, which is a complex community of cell types, I mean, it can be a protein or it can be even a drug, the effect is not going to be selective on one cell type. The effect is going to be on all the cells that are able to sense that signal, respond to that signal. We are looking now at the vasculature, and we're looking now also like potential neural cells, that the neuromuscular junction, to see if there is an effect or if it's a global, basically metabolic signal present in the tissue. We have not tested that yet, but that's what we want. That's the next point. Yes.
That's it. I think I had these important questions. Thank you very much. I think we are almost there with the time. So I really appreciate your time. So thanks a lot, and we'll touch base with you. I'm interested in the conference, so I might want to talk to offline about that sometime.
Absolutely, anytime. Again, we are very excited that we are doing it, and we hope to have as many people as possible in terms of the community that can contribute. I'm happy to talk to you anytime about that.
So,
And thank you for having me, and thank you for doing this. This is an incredible resource. So
yeah,
online, thank you.
sure. We can stop it and we can talk if you have few more minutes.
Yeah, I do. Yeah, thanks.